And our first talk today is from a postdoc, Dr. Leda Lacaria. She is a postdoc at the Institute of Developmental Biology in Marseille, and she is part of the Pierre-François Linné's research group. And her work as a postdoc is focusing on the codynamics of contraction and adhesion in animal morphogenesis. She received her PhD in 2022 from the Aix-Marseille University, and her thesis was titled Cell Mechanic Regulation by Cytoskeletal Dynamics, combining atomic force and optical microscopy under the mentorship of Dr. Felix Rico. And this is what she will be presenting on today, was her PhD work. She is also a very successful scientist, so we're very lucky to have two amazing speakers today. She currently has 5 publications, 3 of which are her own first author publications, which is really exciting. And she has received numerous awards throughout her PhD, such as the Marie Curie PhD Fellowship. And so with that, I'm really excited to introduce both speakers, but we'll start with Dr. Lacaria. So if you want to go ahead and share your screen, we can get started with the talk.
Thank you for the very nice introduction. Hi to everyone. I'm glad today to talk about my PhD project, my PhD thesis on congenic RAS induces cell softening of human mammary epithelial cells via changes in cytoskeleton structure. In simple words, my PhD work is focusing on cell mechanics in cancer, in cancer, so mechanical properties of cancer cells. We know that cancer is a super complex process, but we can simplify it with this sketch. So we know that cancer starts when one cell, so a few cells undergo a genetic mutation, and then they divide without control, and they form the primary tumor. Now, at this stage, the probability of survival are higher because this primary tumor can be removed by surgery, but the trouble starts when these cells start to be able to invade locally the extracellular matrix, to intravasate in the blood vessels, circulate in other area of the body, and then through extravasation form a secondary tumor or a micrometastasis. So already from and this of course simplified sketch, we can understand that is super important. Cell deformability is very important in the process of cancer progression, because the cell becomes able to invade healthy tissue and to go through healthy cells and to deform without any damage. So that's the reason why in literature it's well studied the mechanics of cancer cells. In particular, some pioneers in this field is Leca's lab, and we can observe from their data that comparing normal cells with malignant cells of different cell lines, malignant cells are always softer than normal ones. Here in this graph, we have the distribution of Young modulus. That is a way to quantify cell softness or cell stiffness. So when the Young modulus is higher, the cell is stiffer. When the Young modulus is lower, the cell is softer. And malignant cells are always softer than normal ones. And this is also observed, this phenomenon, in primary cells, so in cells of primary tumors. If we compare normal cells with tumor cells are softer than normal ones. And this is a phenomenon that is common in all cancer cells in different kind of cancer models. Indeed, here we can see an histogram of Young modulus in the case of different cancer, like thyroid cancer, breast cancers, kidney cancers, etc., prostate. And we can observe in blue normal cells, in red cancer cells. And the blue cells show always an higher Young modulus, and the cancer cells instead they have always a lower Young modulus. So cancer cells in all these models of cancer are always softer than the normal ones. So in general, if we take in consideration two cells, a cancer cells and a normal one, normal cells, if we apply a force, an external force on them, cancer cells will deform much more than the normal cells. So cancer cells will be softer than the normal cells, they will be stiffer. And this phenomenon is called cell softening in cancer progression. So now the question is why cancer cells are softer than normal ones. And we know in literature that in general, mechanical properties of cells are affected a lot by cytoskeleton structure and organization. And it looks like that the main role in mechanical properties is that one of F-actin and actin cytoskeleton. Indeed, if we consider like these cell lines, when we use a drug to modify the structure and organization of F-actin, that is cytochelasin D, we can observe always a reduced Young modulus. So when we change the organization, when we disrupt the structure of actin cytoskeleton, we decrease cell stiffness. So now the aim of our work is to quantify the mechanics and cytoskeleton composition and organization of normal and malignant cells, of normal and cancer cells. To do this, we need a very robust model to study cancer, and we used, for this reason, immortalized human mammary epithelial cells called HMLE cells. These cells, we use these cells thanks to the collaboration with the Centre de Recherche en Cancérologie de Marseille, so in collaboration with some medical doctors, experts of cancer. And if these HMLE cells are injected in a mice, we will not observe any tumors or metastasis. But if we modify the same cell lines and we activate the RAS pathway, and we inject these cells in a mice, then we will find tumors and metastasis. So this shows that human mammary epithelial cells are behaving as stem cancer cells when the RAS and MAP pathway is activated in mice. Then, at the same time, we need also a robust method to compare cells, because it's not always easy to compare cells and to have a very robust quantitative way to compare normal cells with cancer cells. To do that, we use cell micropatterning. Indeed, we can control, we can create an adhesive micropattern with a control geometry. In our case, is a bow arrow. And in this way, the cell can adhere just in this area that is where is coated with fibronectin. And the external area in white instead is coated with PLA-PEG that is not allowing any cell adhesion. And thanks to that, we can control the cell shape, so have all the cells with exactly the same shape. And this is a very good technique because it's allowing us to collect all the cells that otherwise will be very different and to do an average map image of all the cells. And in literature, it's known that the cell micropatterning is robust to control not only cell shape, but also to control also the structures of the cytoskeleton in the different areas of the cell. And in Rigato et al., this paper is showing that is a good method to control also mechanical properties of the cells. So we find, thanks to cell micropatterning, we can make more homogeneous and have less differences between the cells and have a robust way to compare them. To measure mechanical properties, we use atomic form microscopy, AFM. The atomic force microscopy is working like an old vinyl player. In a vinyl player, we have a lever with a tip, a needle at the end, and this lever is moving up and down in function of the peak and valley of the disk. And thanks to this movement, we have an electric signal that is the music, converted in the music we can listen to. In a similar way, atomic force microscopy is working. Indeed, we have a cantilever of a size of micrometers with a tiny tip at the end. And thanks to its movement, when this cantilever is touching the surface, we can obtain a topography with nanometric resolution. So the atomic force microscope is more a nanoscope because we can obtain topography with nanometric resolution. At the same time, the atomic force microscope is also a force sensor, in the sense that thanks to this cantilever and recording and cantilever bending, we can really measure forces at piconewton scale. And thanks to that, it's used for single molecule measurements or for measurements of mechanical properties. So combining these two potential of this microscope, using force mapping, we can have at the same time the topography with nanometric resolution of our sample and also the stiffness map of our sample. How we do that? As I told before, so in atomic force microscope, we have a cantilever with a tip at the end, and we can move this cantilever up and down on our cells, that is on the inverted optical microscope. And the bending of the cantilever is recording with a laser and a photodiode. When the cantilever is far away from the cell, the deflection, so the cantilever deflection is 0. But when we approach the cell, we start to deform the cell and the cantilever starts to bending. So here we have an eigenvalue of the cantilever deflection. And then when we move the piezo up, we retract the cantilever from the cell, and then we can measure some addition between the cantilever and the cells, and this bending in the other direction is the addition. And then we have again the bending of the cantilever, so cantilever deflection 0. We can see in this graph in light blue the approach curve, and in dark blue the retract curve. And the point of contact is the point in which the cantilever was touching the cells. And thanks to that, we can know what is the maximum deformation of the cells. So we can convert the piezo eye in indentation. And then since cantilever is exactly elastic like a spring, thanks to the Hooke's law, we can convert the bending of the cantilever, the deflection, in force. So we have our force curve, this graph, force in piconewton in function of indentation in nanometer. And fitting this force curve, we can measure the Young modulus. The Young modulus is the quantity that is giving us information about the stiffness of our sample, the mechanical properties. And if we plot the maximum deformation, the maximum indentation of our cell, we can have also the topography of the cell, or we can plot also the contact point. And if we acquire these force curves in different points of the cell, so we move our cantilever not just in one point up and down, but with a motor in X and Y direction, so we are scanning our sample, we can then obtain a map with many pixels, in which each pixel there is this force curve, and that's how we obtain these two images. One image is the Young modulus of a living cell, here in upper, and then the other is a topographical image of our cell. And we can do this with a resolution that is around 50 nanometer, in which we can really distinguish different structures of cytoskeleton. So thanks to this method that is called mechanical mapping, we can correlate on the very same cell at the same time cytoskeleton structure with cell mechanical properties. And we can do that on living cells. These are for an example on living fibroblasts, but we can do exactly the same on our living HMLI-RAS cells. And this way we can compare normal cells and cancer cells. So thanks, as I told you before, we are using also micropatterns. So what we are doing is that we obtain this topography and young modulus map per each cells for 25 cells. And these cells are micropatterns, so they will have exactly the same shapes. And thanks to that, we can take all these cells and average the images in one image, only one image. And here, this is the average image, the topographical image of HMLI cells. And this is instead the young modulus image, so the stiffness map of 20 cells, the HMLI cells. And we can do exactly the same with normal cells and cancer cells, so comparing HMLI and HMLI-RAS. What we observe if we do that, we have on the top HMLI cells, normal cells, down cancer cells, and we can see first of all that in the young modulus map in HMLI-RAS, the cell is much softer. If we look at the histogram, we have a total shift of the histogram, so the cells is overall much softer than the normal ones. So RAS activated cells, cancer cells, is softer, and this is in agreement with what was described in literature. On the other side, if we compare also the topography, we can see the HMLI-RAS cells are flatter than normal ones. Now, after this, we want also to understand how the mechanical properties of cells of cancer cells and normal cells are affected by the addition of the cells on the substrate. So we want to know if this difference between cancer cells and normal cells is due to differences in pre-stress and in addition of cells. To measure this, we use traction for microscopy. So we make some polycramide gels that are known for their elastic properties. We coat them with some fluorescent beads, and we acquire the images with the position of these beads with the cells and without. Thanks to this comparison, we can calculate the displacement, and from that, we can compute the traction forces. So the forces that the cells is exerting on the substrate while is adherent on the substrate. We have done this with our HMLI-RAS cells. So on top we have normal cells, and on the bottom we have cancer cells. And if we look at the total force, we cannot see a statistical difference, and also we cannot see a statistical difference in net contractile moment. But what we observe is that if we measure the dipole, we have the distribution in cancer cells, so HMLI-RAS cells, is much more polarized. The dipole is giving us an information about how is the main direction of the tractions in the cells. So if we have two distributions that are suggesting that the traction forces of HMLI-RAS are more polarized. So there are more cells with one direction and other with another one, while in the HMLI cells is more random. Then we want also to correlate, as I told, the mechanical properties of these cells between and compare normal cells with cancer cells and correlate this with the organization of the cytoskeleton in the cells, on the cytoskeleton elements. So we looked in our cells at actin, but also at vimentin and cytokeratin 5. Actin because it's known to be to have a very important role in regulate mechanical properties of cells. But vimentin and cytokeratin also because they are known for their important role in epithelial mesenchymal transition, and so our hypothesis is that they can have a role in cancer progressions. So here on the top, you can see the fluorescence image of our pattern with fibronectin GFP, and then we have a bright fields image of H-Ras. And then if we have the actin, vimentin, and cytokeratin averaged images of 25 cells. If we compare the average image, we cannot see a big change in the intensity between the two cell lines, and also seems that the organization and distribution of the cytoskeleton is quite similar. But we can normalize the actin or cytoskeleton image we have, the median fluorescence map, with the thickness of the cell, since we know thanks to the topography image of the cell. If we do that, we kind of measure the density of the actin or of the vimentin or of the cytokeratin inside the cell. Because when we obtain the median average, the average image of the fluorescence signal is a Z projection of all the volume of the cell. If we look then at the density of different cytoskeleton elements in the two cell lines, HMLE and HMLE-RAS, we can see that there is not a big difference in F-actin. If we look at actin normalized for the thickness of the cells, but instead, if we look at the map of vimentin normalized by the thickness and cytokeratin 5 normalized by the thickness in HMLE-RAS, the value is much lower. So this is suggesting that the density of vimentin cytokeratin 5 are much lower in RAS cells, in these tumorogenic cells. So this suggests that the cytoskeleton in HMLE-RAS is less crowded because vimentin and cytokeratin are less dense. And then we can observe also that vimentin is more concentrated around the nucleus in HMLE-RAS cells. Then, if we compare these images with the Young modulus, we can see that the actin map, the actin density map, correlates very well with our Young modulus map. And this suggesting that the stiffness of the cell that we measure correlates very well with the distribution of actin that is much higher in the periphery of the cells and lower in the perinuclear area of the cells. And this is something that we can observe in both cell lines. So then we want to understand if this difference in mechanical properties, so in Young modulus between normal cells and cancer cells, so HMLE-RAS, can be due to a different orientation of the filaments of F-actin. To do that, we use polarization resolved fluorescence microscopy. That is a technique that was developed in Institute Fresnel by Manos Mavrakis and Sophie Brasselet. So we know that light can have different polarization. Can have a linear polarization, circular polarization. Now, if we consider the linear polarization, we can change the direction of the polarization, changing the angle alpha. And if we change the polarization, the angle of polarization of our laser beam when we do some fluorescence microscopy, and we can obtain different image in function of alpha, then the fluorophore of the protein we are interested inside the cells, for example, in our case, F-actin, the fluorophore attached to F-actin, in this case, phalloidin, will behave as a dipole interacting with the polarized light. And we can obtain this graph of in function of the alpha, of the angle of the polarization. So thanks to this graph, we can measure the direction of the dipoles, so of the actin molecules in ensemble in our cell and have the main direction. So that is actually the row, but at the same time we can also know if what is how is the if these dipoles are ordered or disordered. So how the standard deviation of the direction of these dipoles, measuring psi, that is the this solid angle. So in a more ordered structure, we will have a psi that is smaller because all the dipole, they will be similar. The direction of the dipole will be similar. Instead, in a more disordered structure, we will have a bigger psi because we have a big range of direction of dipoles. If we use this technique in our cell lines, we compare here HMLE, RAS. So if we look at the orientation row, HMLE cells and HMLE RAS have a very similar distribution. So this means that the organization of the actin filaments is very similar. The orientation is at molecular level of the ensemble is quite the same. But we observe that instead the psi value is different. Indeed, in cancer cells, so HMLE RAS, the F-actin at molecular level is much more disordered. If we take all these results together, here we have the image of all the data I presented until now. We are correlating cell mechanics, that is thanks to IFM and traction for microscopy. So we know we measure the cell deformability, we correlate it with the cytoskeleton pre-stress and the adhesion of the cell. Then at the same time, we have information about the cytoskeleton composition, structure, and organization. We look at F-actin, cytokeratin 5, and vimentin. We observe a difference in the two cell lines of vimentin and cytokeratin 5 density, a difference in the thickness of the cell, and most of all, a difference in the F-actin molecular order. So to conclude, oncogenic RAS expression induces a wall cell softening, and this is in agreement with literature. More spread phenotype, an higher tractions polarization, a lower intermediate filament density, and a more disordered actin filaments on molecular scale. So we can say that the molecular order of actin may be a signature of the softness in cancer cells, and we speculate that probably the density of having a density of intermediate filament is lower, make a less crowded cytoskeleton inside cancer cells, and this can affect may affect the orientation of actin, and maybe affect the mechanical properties indirectly. But this is just speculation, but what we show are these results. So I want to thank you for your attention and say a special thanks to my supervisor, Felix Rico from the Dynamo Lab in Marseille, my co-supervisor, Loic Legov, and all to our collaborators, Xavier Trepat and Raymond Suner in IBEC in Barcelona, Manos Mavrakis, Sophie Braslet in Suffrenel, and also the group of Emmanuel Sharaf-Joffre and Christopher Guinestière in CERCEM. Thank you very much.